Hello everyone, welcome to Sankalp Learning Classes. Let's study about the mutation. Okay, what is mutation? Mutation is the phenomenon means which results in the alteration of DNA sequences and consequently results in the changes in the genotype and phenotype of the organism. So here mutation means change or the alteration in the DNA. So the DNA sequences that changed because of the some other factors, and the change in the DNA sequences that leads to the change in the genotype also and the phenotype. So it also causes the changes in the phenotype of the organism. And in addition to recombination and mutation is another phenomenon that leads to the variation in the DNA. So the mutation which leads to the variation in the DNA. So what are the reasons for the mutation? Loss, that is deletion or the gain. So sometime, for example, adenine, guanine, cytosine, adenine, guanine. So these are the nitrogen bases that are present on the DNA sequence. So the addition of the some other sequence or the deletion of some other sequence on DNA that result in the alteration in the chromosome. Okay, so since the genes are located on the chromosome, alteration in chromosomes result in the abnormalities or the aberration. So that also leads to the aberration. Chromosomal aberrations are commonly observed in the cancerous cells as a point mutation. As a point mutation, for example, sickle cell anemia. So here, what is mutation and how it occurs? It is because of the loss of chromosome also, loss of the DNA sequences or the gain of the DNA sequence. So that is the reason for the mutation. Then mutation also arises due to the change in the single base pair of DNA. This is known as the point mutation. So just imagine this is the DNA sequence. So if there is this is the normal A, T, C, G, A, T, and complementary sequences are present on the other strand. So this is 5 prime to 3 prime. And this is 3 prime to 5 prime. Okay, so the change in the DNA sequence. So A should be there, adenine, guanine, adenine thymine should be there. So here adenine, some other guanine is there. Okay, so change in the single base pair. So change in the single base pair of DNA. So that also leads to the mutation. So that type of mutation is called as point mutation. That is the sickle cell anemia. So deletion and insertion of base pairs that cause the frame shift mutation. So what is this frame shift mutation? So example for the point mutation, that is sickle cell anemia. And the causes of mutation. Mutations can be caused by the different agents and are known as mutagens. So what are mutagens? Very important. The mutagens are the agents that are caused by the mutations can be caused by the different agents, and the agents of the mutations that are known as mutagens. Mutagens can be physical or the chemical. So physical mutagens, example like high temperature and the radiation and UV rays, alpha rays and gamma rays, X rays. So these rays that cause the change in the DNA sequence, DNA base pairs. So these are called as physical mutagens, and chemical mutagens like mustard gas and nitrous gas are the example for the chemical mutagens. So here what are mutagens? Example for the physical mutagens and example for the chemical mutagens. So this is very important in this topic. And the types of mutation. Already we know the point mutation. What is the example for the point mutation? That is the sickle cell anemia. And the one more type is the frame shift mutation, right? So here the point mutation, that is due to the change in the structure of gene. So this type of the mutation normally alters the information covered by gene. So for example, the gene is changed means the different type of protein is synthesized. When the protein is different means that alters the uh, phenotype, that provides some other expression, right? So in this, there is a mutation is only one nitrogenous base of the triple codon and thus known as point mutation. So in the point mutation, only one base pair is changed. Okay, so only one pair is changed in the triple codon. So that is the example for the point mutation. And the second type, that is the frame shift mutation. So what is this frame shift mutation? So here in this, there is an addition or loss of more than one nitrogenous base. So there only one nitrogenous base is changed, that is the sickle cell anemia, right? But here more than one nitrogenous base that is changed, it results in the shift of the nitrogenous bases and reading of the codon is affected. Hence, the protein formed is different or may not be formed at all. So, for example, AUG is there means, so here there may be a change in the AUG also, or sometime AU is changed. Okay, so this total AU only changed. So there is a change addition or sometime AAUG is added or AAAUG is added to the AUG. So here more than one nitrogen bases that is added to the normal gene. So that is called as the frame shift mutation. Sometime the 3 AUG is removed means so that the total the protein is only not produced, right? So the complete shift of this the gene takes place. AUG next, UAG is there or some AAA is there. So this only shift, this so this codon is only removed, so then it causes the frame shift mutation. Thank you.